Good morning. Hello, everyone. How are you today? Good. I won't ask about yesterday. I'm going to cross my fingers that it went well. I haven't seen the outcomes yet. Have you? Have you seen the scores yet? Are they released? Okay. You smile. Hopefully that's hopeful. So are things kind of making a bit of sense to you now? With Dr. Thung's lecture yesterday, I can't remember what it was on, and even his lecture today, are you seeing connections between things that we've talked about? He dealt with, today was, what was today? Apoptosis, right. So you would have, he brought up phosphatidylserine, right? Yeah, okay. And involvement of, he would have talked about phagocytosis, yeah? So phagocytosis is going to require, do you know the names of some phagocytes? What do you say? You're like what they said. No, tell me, I want to hear you. Why do you think I come back here? Not because you're all pretty, because you are, but you know, still. I hear them all the time, so that's why I come back here. So you agree? Names of some phagocytes? What are names of some phagocytes? Anyone? Macrophages, osteoblasts, good, good, good. And for them to do their work, you need to have rearrangement of the cytoskeleton, right? I'm not sure if that would have come up with him, but you recognize that, yeah? So I don't know if you've seen pictures of the phagocytes. When I say like the big hug, it literally is like that. The membrane becomes deformed and it forms, so let's say I'm the cell kind of round, and then it kind of a part of the membrane deforms and it goes around that cell that has died by apoptosis and basically takes it into itself. So to form these arms, you got to rearrange the cytoskeleton, right? What aspect of the cytoskeleton are you rearranging primarily? The cytoskeleton. That's it. The actin filaments. Polymerizing and using, what do we need for that? Were you in my tutorial group the other day? No. No? Okay, good. So what do you need for actin polymerization to form the branches? So the energy requiring molecule, the energy molecule that you need is ATP or GTP? ATP, A for actin. But remember actin branching? What do you need for the branching to occur? WASP and ARP23. Okay, good, good, good. So all of these things that we're doing, I don't know. Sometimes they might seem like mishmash, disparate topics, but they interconnect. So cell adhesion today and the extracellular matrix, another fun topic. Don't laugh at me, it really is fun. Well, first of all, the basics. The textbook is the same as the one before, where you have to, that annoying thing where you have to click on a lot of the individual links. But what are we going to cover? We're going to talk about different types of junctions. And I'll tell you what junctions are, but these points of connection that mediate adhesion of one cell to the cell next to it. We're going to talk about the major proteins that you find within the junction that actually do the work. We'll touch on matrix metalloproteinases, MMPs. Any of you who ever done work with stroke patients might maybe, or at least maybe you've heard of TIMP. Anybody? Anyway, okay. And then we'll touch a little bit on the extracellular matrix. Not a whole lot. More of that gets covered in the next course when you come up on anatomy. So, and I'll tell you along the way how it ties in with what Dr. Thung and maybe some of my other colleagues have done so far. So let's start off with the first two objectives, the types of junctions and the proteins. Yes, I'm going to tell you up front, more protein names to remember, okay? And the types of proteins that mediate these interactions. So first of all, let's do the basics. Our a lot of the times we talk about epithelia, and I know when, I think, if I can remember, when I was learning this years ago, I thought first of, I mixed up epithelia and epidermis. They're not completely different things. They are similar, but they're different. So epidermis, we're talking about the outer layer of the skin. And epithelia really, singular is epithelium, the layer of cells that lines the outermost surface of whatever tissue you're talking about. So if you think of the stomach, that layer of cells that is facing the lumen of the stomach, the space where the food goes, that layer of cells, that's the epithelial layer of the stomach. Think about our blood vessels, you know, about arteries and veins and capillaries and so on. Blood vessels are lined by, the wall of blood vessels is made up of a certain type of cell, and I actually do want you to remember this. So those cells are called endothelial cells. So endothelial cells, if you know it already, great. If you don't, you might want to take a note of it, are the cell types that make up the walls of our blood vessels. So you know about enterocytes, epithelial cells of the intestines, endothelial cells, epithelial cells of the blood vessels. So skin, body cavities, etc. So that's what we mean by epithelia. That's the first word to add to your vocabulary if necessary. What do epithelia do? A whole bunch of different things. So I've been using the intestines a lot as an example. So those intestinal epithelial cells with the microvilli, absorption, the epithelial cells here, barrier function. When I say barrier function, what do you understand by that? Protection from what? Anything external that's not supposed to be in the body, even things that maybe are supposed to be in the body. When you put a drop of water on your hand, does it slowly absorb in? If it does, go to the doctor. Okay? It shouldn't. So skin forms a barrier, both from the bad things and the good things that you want to regulate the intake of. Bugs, bacteria, etc. So different functions. So epithelial cells are referred to as being polarized. So you know about your polarized lenses, but it's a slightly different type of polarized. Polarized meaning they have, I'm going to say it in layman's terms, they have a top side and they have a bottom side. And I think I mentioned this briefly at some other juncture. I'm having a little deja vu again. So let's take the picture at the top. These simple columnar cells are examples of polarized epithelial cells. So the top part, quote unquote, the top part is referred to as the apical surface. Why am I not getting colors? Huh. Okay, I only have black. All right, whatever. So the top side is known as the apical surface, and the apical surface of the cell is always the surface that is facing the lumen of whatever organ we're dealing with. So it's going to be the apical surface of the blood vessels is going to be, of the blood vessel wall, the endothelial cells, is going to be the side of those cells that is facing the actual blood. The apical surface of the enterocytes, or intestinal epithelial cells, that's going to be the side of the cells that's facing the food that you've eaten and is being digested. Airways, apical surface of those epithelial cells, the surface that's facing, and your esophagus, the one for food and the one for air. What are they called? Esophagus and trachea, right? Thank you. You get the point, right? So the apical surface is the side facing the lumen. There's a lateral surface, and the lateral surface is the side that is sitting adjacent to the next door cell. And then we have the basal surface, the bottom surface, so to speak. That's going to be the surface that is sitting on our extracellular matrix, depending on where it is. Maybe it's facing interstitial fluid, bloodstream, whatever, okay, on the outside. So those terms are important because, you know, in a question and it specifies apical surface, that should direct your mind to certain types of things, because only certain types of proteins, for example, are going to be found on the apical surface. Different ones are going to be found on the basal surface, and we'll talk more about that. And the cartoon below is just showing you similar things. So our basal cell surface is sitting on a basement membrane, which is not stated anywhere in these pictures, but okay, it's all right. The basement membrane is synonymous to the basal lamina. Now, students initially get confused about that term because membrane, right? We did membranes. Membrane is a part of the cell. Not in this case. In this case, the basal membrane is not a part of the cell. It's a part of the extracellular matrix. Somebody gave it that name to just confuse the heck out of us, but we'll get there, right? So, yes? Yes, and we're going to talk about that later in this lecture. So, basement membrane, basal lamina, same thing. Somebody first identified it using, I don't know, maybe electron microscopy, and someone else identified it using something else, and they came up with different names and eventually realized, okay, they're the same thing. And you still find both names being used in the literature. So I need you to know that they are the same thing, and that the basement membrane, even though the word membrane is in it, it's not a part of the cell. Initially, that is going to trip you up. Make a note if you need to, asterisk, whatever. The basement membrane is a part of the extracellular matrix, not a part of the cell itself. And then below that is the connective tissue. So you're seeing here the basal lamina, this, and the basal lamina, I'm sorry, I don't have another color, this, for whatever reason. But, so this thing here is the cell. And then this dark layer just below it, if you can see where I'm drawing, even though I don't have colors, that is the basement membrane, aka basal lamina. So it's a very thin extracellular matrix layer. And then below that is our connective tissue. And in anatomy, they'll tell you more about connective tissue, loose connective tissue, et cetera, et cetera. Okay, so this is a general architecture that we're going to be referring to over and over again as we go through this next couple hours. So the title is cell adhesion. Why do we need it? What's that? Why do we need adhesion? Okay, communication. Well, we can communicate long distance. We don't need that to talk to each other. Barrier? Barrier? Barrier function is a little bit different from adhesion function. I'll tell you more about the difference a little bit later. That's why you'd want cells to be side by side. But why do we need cells to adhere? To anchor. Yeah, I like that word. You said? That's important. That's one of the side effects, that it will allow that to happen. He's talking about transfer of nutrients. But that anchor word, that's it. So once upon a time, apparently, so they tell me, we started out as a single cell. And some of the stuff around us is still single cells. But the moment we graduated, the moment we ascended into multicellularity, adhesion became important. Can you imagine, have you seen those movies? I can't remember which one it is. Might be that weird one with Leonardo DiCaprio, where the people were kind of yeah, I'm not sure if it's that one, but there were times when people were kind of like floating away as little atoms, molecules. I think that's the wrong movie, I can't remember. Anyway, the point I'm making is, multicellular beings need adhesion to be multicellular beings. Otherwise, we'd be all just a floating mass of cells and not being able to do all the things we do. Yeah, okay, so we won't go into the sci-fi movie, but hopefully you get the point. What do cells adhere to? Say again?
The sentence you said before you said we're not going into sci-fi, what was that sentence you said?
I don't remember. Can someone help? What did I say? Oh, okay, yeah. Multicellular beings, yeah. No problem. Thank you. Thank her. So what do cells adhere to? Again, it's a very simple question, but these are the basics we're starting with. Other cells, yeah. The adjacent cells. Not the cells way down there, the adjacent cells. Anything else? The matrix. That matrix, not Keanu Reeves, but this extracellular matrix, okay? So those are the things that cells need to adhere to, in essence. Some types of cells are not adhesive at all during their lifespan, at least not in a normal lifespan. Example? Our blood cells, erythrocytes, even white blood cells, okay? They're not supposed to. What would happen if our red blood cells, for example, started sticking to the folks next to it running through the blood vessels? Blood clots, for example, block up vessels, causing us to get a stroke, what have you. Don't write that down. So it's important for some cells to not be adhesive. And when we talk about the adhesive molecules, think about, okay, so these types of cells are not going to normally express the genes that code for these adhesive proteins, they're proteins. They're not normally going to be expressed on our red blood cells, for example, because they should not adhere at any point during their lifespan. Okay, so getting to these junctions, as I mentioned briefly earlier, a junction is simply a point of contact between two adjacent cells or between a cell and its the underlying extracellular matrix, the basement membrane. So they mediate cell-to-cell adhesion and they mediate cell-to-ECM, ECM being extracellular matrix. Okay? They're going to regulate polarity. So when I talk about polarized cells, how do they regulate polarity? Think about so let's say this here is a junction, okay? These are all junctions. And remember I said, let's take our intestinal epithelial cells, for example. The enterocytes, they're going to have a protein on their apical surface to import glucose from the lumen. What do we call that protein? SGLT1, exactly. So remember back when we did membranes and I told you about the fluid mosaic model. Remember that? And we're not just static here. Some proteins can breathe a little bit, you know. I can end up being over there as a protein. So what's to stop me from not going over that side or going all the way over there? Or headed back that way, depending on how this cell is shaped. If SGLT1 ends up on the basal surface, create this picture in your mind. If SGLT1 ends up on the basal surface of our enterocytes, we have a problem. What's the problem? Glucose would not be able to be absorbed because you need them in the place where the glucose is, which is the lumen. Right? So it's important, even though they have some degrees of freedom, we don't want them going crazy and going all over the place, right? The junctions are important for maintaining them within a certain geographic region, if I can say it like that. So in that way, they regulate cell polarity, keeping the cells that should be on the apical surface on the apical surface, and similarly for the other proteins on the other surfaces. We talked about, someone mentioned communication. Well, I'll dig into that a little bit more later. But yes, they're involved in cell communication. Yeah?
So when you're referring to cell polarity, you're referring to, not just the charge, you're referring to basically the geographical regions of the cell and them being different.
Yes, exactly. So he said, when I'm referring to cell polarity, I'm not necessarily referring to a charge, but referring to different geographic regions of the cell that, well, as the word just said, they need to be different because they perform different functions. And it's funny you say that about charge. I was going to just make the analogy with the battery. You know, you have the positive end, the negative end of the battery, and you can't flip them around. They're not going to work like that. Well, again, with our enterocytes, if you flip that cell around and put SGLT1 on the basal surface and put GLUT2 on the apical surface, it's not going to work. GLUT2 cannot take up glucose by facilitated diffusion from an area of lower glucose concentration, which is the lumen, to an area of higher concentration, which is the enterocytes. And remember we said enterocytes, as strange as it seems, they have a really high glucose concentration. So exactly, yeah.
Just to clear two things up, the apical surface, that part of the cell is where the lumen is, or is it with the bloodstream?
It's facing the lumen of whatever
Now,
organ.
that's intestinal When you're talking about a cell, and you mention the word lumen, it's the opposite of bloodstream. If it's a stomach cell, it'll be inside the stomach. If it's a anterior site, it'll be inside the. Is that right?
Yes and no.
Okay.
So his question is about the word lumen and what it means exactly.
Because I've seen that in a.
So the lumen is always, so we even use the word lumen when referring to the endoplasmic reticulum. It's the inside part, the space. So you just mentioned blood as an example. If we're talking about the blood vessel wall, what were they called? Endothelial cells that make up the blood. So if those are the epithelial cells we're talking about, then the lumen is where the blood is. You have that picture in your head? Because that's the space. Think of the blood vessel as a hose, and where the water would run through is where the blood is running through. But the wall of the hose, those are our endothelial cells. So the side of the cells facing the space within, that space is where the blood is flowing. But if we're talking about our enterocytes, our intestinal epithelial cells, so up here is the lumen, where you have the food, etc., and the water you drank, and yada, yada, yada. And down here, below the basement membrane, I'll show you a picture later. Below the basement membrane, we have interstitial fluid, and eventually we have some blood vessels running down here. So here, the side of the cell that faces the blood vessels, if you will, that's the basal surface. So it depends on the tissue you're talking about, but no matter the tissue, the common theme is whatever side of the cell is facing the space within, that is the apical surface. So use any example you can think of. Blood vessel, intestinal lumen, airways. Just 1 second, I saw this hand.
With the gap junctions, right, or junctions, do they only generally attach from cell to cell, or do they attach like cell to extracellular matrix?
So he's asking junctions in general, do they attach only cell to cell, or do they do cell to ECM? Both. Oh no, the question went over your mind. Whatever side of the cell is facing the space within, that is the apical surface. And that space within, in quotation marks, depends on what organ we're talking about.
So like in the example of the blood vessel, let's just say the erythrocytes are in there. So this side is the apical and here is the basolateral.
Okay, so she's asking about blood vessels. Let me draw. Hope it helps you more than it hurts you. So this is where our blood is. Plasma, red blood cells, etc. These two lines are actually this. We draw a line because it's convenient, but this is what that blood vessel wall actually looks like. So in this case, where's the lumen of the vessel? It's going to be here, right? This is the space within. And therefore, the apical surface is going to be at point number 1 or at point number 2? At point number 1. Is going to be the basal surface of the cells. Does that help a little bit? And draw a stomach, draw whatever you want. Same. Facing the interior of the stomach, apical surface. Did I see another hand? There are 3 major classes of junctions. The lady at the back said anchoring. We have occluding junctions. Someone asked about barrier. So that fits there. And then someone else said communication. These are the 3 major classes. Let's talk about them. So they're different, and I'll explain the different characteristics as we go along, how anchoring is different from occluding. Technically speaking, the anchoring junctions are the ones that have the adhesive proteins. And I'll explain that further. So let's talk about those different types of junctions, A, B, C, D, down there. The first type is the adherence junction. I put the old name in brackets, but less and less I see zonular adherence. Most times you're going to see the term adherence junctions. So let's look at these cells. We have 3 cells on the right-hand side, right? Cell number 1, cell number 2, cell number 3. The adherence junctions, and notice where the lumen is. Why don't I have colors? I don't get it. Okay, notice where the lumen is. So these look like intestinal epithelial cells. They have microvilli and all of that. So this is where our food would be. And notice the basal surface of the cells. So our adherence junctions, the note says, found near the apical surface in the epithelial cell layer. Meaning, 1, 2, 3, that's a part of our epithelial cell layer. They're going to be found near the apical surface, not down here. That would be near the basal surface. Their function, adherence junction, so remember, a junction is just a point of contact between adjacent cells or the cell and the ECM. So adherence junctions in particular, one of our anchoring junctions, their function is to hold cells to their neighboring, to their adjacent partner. And here we're making the distinction between cell-to-cell adhesion and cell-to-ECM adhesion. This is something that you need to always pay attention to. Make great exam questions. So adherence junctions, cell-to-cell adhesion, and the major protein within that point of contact between adjacent cells, those proteins are called cadherins. They're a family of proteins. They're in red, so hey, kind of important, right?
So are these the proteins that link to the intermediate filaments, so it helps in the structural integrity of the cells and also the gap entry?
So he's asking if they link to intermediate filaments for structural integrity. Some types do, but not this type. Okay, I'll tell you which types do. So point of contact, cadherins are the name of the proteins. The cadherins are sitting in the plasma membrane, okay, and that's how they're able to make contact with the adjacent cell to perform their adhesion function. So here is, I think, tomorrow, or have you seen this already? Has Dr. Thang already told you a little bit about embryogenesis? Tomorrow. Okay, so when he tells you about this tomorrow, well, you don't need to know anything special for me. I just want to bring over the adhesion application here. So let's talk about the birds and the bees. Sperm meets egg. They come together, fuse, sperm goes in, and we have ta-da, zygote, right? So in essence, we have one cell that is going to become a baby. Along the way, that cell divides into 2, and then it divides into 4. You don't need to write this down. This is gonna, you will have to in anatomy. And that 4 becomes 16, and eventually we stop calling it 4 cells or 8 cells, and we start calling it a blastocyst. Anyway, the point I'm making, unless you want twins or quintuplets or whatever, adhesion molecules are required to keep those cells together every time they divide. Okay? And so we're going to move eventually from the ball of cells, and we're going to differentiate, and you'll get the whole story about that. But that ball of cells will eventually form brains and viscera and whatever and whatever. But just know that during that process of embryogenesis, adhesion is critical. Without it, there is no baby, or you end up with 20 babies, and that's not cool. So this is what keeps those cells together as they divide. And so notice here, they're telling you about the adherence junctions. I'll tell you about the tight junctions a little bit later, between all of them. This is just a pretty picture to kind of show you, so you're not responsible for most of the stuff in here. I'll tell you the only one you're responsible for along the way. But remember I mentioned it's a plasma membrane protein. So cell number 1, cell number 2, I'm in my plasma membrane right here, but there's a part sticking out outside, and that's the part that's going to make contact with the similar region of the cadherins in the cell next to me. And that's how the adhesion happens. Okay, so similarly down here, you don't need to know about most of that. I'll clue you into that in a while. Okay, so it's our cadherins. So this is one cell, this is another cell, and these are the cadherins sticking in the plasma membrane. And so this is what they're doing to effect adhesion, basically. But just on their own, they're not that strong, right? So inside the cell, remember they're plasma membrane proteins. Inside the cell, they're going to be connected to a part of the cytoskeleton. And that just helps to strengthen the interaction. So that if you give, you know, it's like a tug of war and you have one person on a side. You have multiple people, that provides more stability. Then we have a real competition, right? Then we have more strength, stability of that adhesion interaction. So for all of these that I'm going to tell you about, they all have a cytoskeletal partner. In the case of adherens junctions, the cytoskeletal partner is, so remember I told you in the very beginning, I put these blanks in here because sometimes I know that, especially the start of some of these lectures, can be a little bit of a snoozer, so this is just to keep you awake and active. And so actin is what goes in here, okay? Now, the adherens junctions are near the apical surface. There's another one, another type of junction below them called the desmosomes. We're going to talk about those in more detail in a little bit. The desmosomes are actually important too. So yeah, these folks are up here, but with the desmosomes just below them, it actually strengthens the adhesion between these two adjacent cells. So that's all that next note is saying. If you have just adherens junctions and you have no desmosomes, those two cells are more likely to be ripped apart than if the desmosomes are there. So it has stability.
And is that where blisters might form? Is that cell you see on
There are different types of blisters. She's asking about blisters, and we're going to talk about those in a second. But yeah, there's implications for blistering. There are. Okay, so when I say help resist mechanical stress, is that a clear term? Mechanical stress, any type of external force being put on it? Okay, all right. So, flashcard, I have it like this, you have it in a different form. What's the answer? Cadherens, exactly, exactly. All right, and throwback, review, integration. What's the answer? Who says non-secretory? Nobody brave enough? You're all smarter than that, that's why. Okay, good. Why secretory? Because they're plasma membrane proteins, exactly. Well done. Okay, so speaking of the desmosomes, let's go to talking about those. So we talked about the adherens junctions near the apical surface. We're going to talk about the ones just below them now, macular adherens. Desmosome is the word that you're most likely to see. So we talked about their location. By the way, these things are not interchangeable. You will not find an adherens junction sometimes mediating cell to ECM interaction, never. Always cell to cell, same with the desmosomes.
Just a quick thing on that picture so I know what I'm looking at. When you, the actin, is it the red or is it the lighter green?
Ah, okay. So this one isn't actually showing you the actin necessarily, but if it was, it would be somewhere around the red. The green part here is that plasma membrane protein actually mediating the interaction.
Because it says that it connects the actin filament bundle in one cell with that of the next. So technically, the actin from one cell connects to the, it's not showing it there though, but it is, right?
Correct, correct. So it's easier to see here. So there's a mishmash of things in between, but notice where the actin filaments are. And in the adjacent cell, you would have the actin filaments down here somewhere also. And so that's what I mean. It's like the cadherens are in effect bridging these with these. That's what they mean by that. When they,
What is actually touching? Is it the cadherens?
yes. He asked what's actually touching? Yeah, just the cadherens.
The actin is not touching.
Correct. They can't, right? because I'm inside my cell over here, and that set is inside its cell over
Right,
there.
so since it says connecting, I just wanted to know if they touch or not.
No, bridging.
All right.
Yeah. Ask as many questions as you need to, because this stuff can take a while to conceptually settle in your brain. So the adhesion molecules, the actual proteins that are mediating the adhesion, that are responsible for adhesion within our desmosomes, they're desmosomal cadherins. So, okay, I said cadherins before, now I'm saying desmosomal cadherins. Cadherins are actually a huge family, and we're distinguishing these members of the family. So, for example, on an assessment, if you see cadherins, it's not going to refer to desmosomes, because desmosomes, the proteins there, we always refer to them as desmosomal cadherins, to be specific. As a matter of fact, in some texts that you read, or maybe even on a question, you may not even see the term desmosomal cadherins. You might see desmoglein, you might see desmocolin. And you have to know that those names are examples of desmosomal cadherins. For the adherens junctions, the cadherins, they have names also, but thankfully, you're not required to know any individual ones. But these you are, okay? Desmosomes, so our adherens junctions are going to bind to our microfilaments. Anybody looked ahead? Desmosomes, what do they bind to? They're going to bind to intermediate filaments. I'm going to abbreviate IF. Remember our cytoskeleton, intermediate filaments? What are some examples of intermediate filaments? Keratin, lamins, neurofilaments, etc. Well, the lamins, no, because they're in the nucleus. But here, we're binding to something in the cytosol, right? So some type of intermediate filament. In desmosomes, even the intermediate filaments have their own special names. So you might see placoglobin and desmoplakin. When you look at the textbook, you'll see those names. So the names of some of the intermediate filaments that desmosomes bind to. These are the two that you'll most often see. Okay, those are names of intermediate filaments. And they are in your textbook. Intermediate filaments. The picture, I haven't gone through it in detail, but I've put a red box around the part that's relevant on each slide, okay? So, how are we doing? Okay. Okay, so again, similar to our adherens junctions, strongly adhesive. Where do you find them? Tissues subjected to significant mechanical stress. And I've put a few examples here. Myocardium, the heart. The heart is constantly that we need desmosomes in between those cells, and adherens junctions also. Okay, this is putting mechanical stress on the walls constantly. So they're important there. When you're having a baby, those walls, significant mechanical stress. Defecation, significant mechanical stress. Blowing your nose even, right? So all of those cells in those epithelia, and even non-epithelial cells, they're going to have these between them.
When you say defecation, you mean the external sphincter?
The anal sphincter. So this is an FYI, okay? So you don't need to make any notes here, but just to give you pretty pictures to connect things to. In this condition, pemphigus vulgaris, it's a rare condition, but examiners love it. So in this one, have you heard of autoimmune diseases? where a part of the body basically attacks itself. And again, you do not need to write this down, but just giving you an example. In this condition, our own antibodies are attacking a part of the desmosomes. So it's like a pack of cards. If you pull out one from in the middle, the whole thing comes crumbling down. So these patients, they tend to have skin blistering, characterizes that particular pathology. So what you're seeing there is a bunch of blisters that were close to each other that eventually ruptured. There's another one called epidermolysis bullosa simplex. Look familiar? Okay, good, good, good. So we talked about that a little bit in cytoskeleton. And in this condition, there's actually a mutation in one of the keratin genes. So where does keratin come into play? We have the desmosomal cadherins at the plasma membrane, but they're bound to some part of the cytoskeleton, right? In this case, keratin. So there's a mutation in the keratin gene that hasn't formed, I assumed its normal structure, and the entire thing is affected. So it's usually identified at birth. Surfaces that are subject to mechanical stress, so palms, soles of the feet, that sort of thing, skin blistering. There are different types of EB. This is not the most severe type by any means, but it can be annoying to say the least. CF means cross-reference. I feel old. It's an old abbreviation we used to use. So he asked if it's treatable. Because it's a genetic mutation, you can't go in and say, okay, you're deficient in vitamin C, take more vitamin C, it corrects it. Gene editing hasn't gotten to the place, I think, where they have been able to correct it. But there are therapies, and a lot of the times you're going to start out by treating the blister, because if that gets infected, you have a whole other set of problems there. Good question. Okay, so I just put this here. This is directly from the cytoskeleton set of slides, and I just highlighted the part that's significant to the topic we're doing right now. Okay? Okay, so what have we just gone over? Cytoskeletal attachment for adherence junctions is actin, good. And for desmosomes, we just said that, intermediate filaments. In some cases, actually, the keratin in epidermolysis bullosa simplex. Sometimes it can be in the placoglobins, etc. Okay, all right. So the hemidesmosomes. Remember, we're doing anchoring junctions, and we just talked about a couple that keep cells adjacent, and their adjacent neighbor adhered to each other, right? Like this. Here, cell to ECM now. And the first one is hemidesmosomes. Hemi means what? Half, yeah. So I think they discovered desmosomes first, and it looked like this little spot, like a circle. When they discovered hemidesmosomes, it looked like a half circle, so they named it as such, but that's nothing that you need to, that's neither here nor there. So hemidesmosomes actually perform a very similar function as desmosomes, but they're connecting the cell to the extracellular matrix. So a couple of different words we tend to use here. So again, strong adhesion between the basal surface of the cell and the underlying extracellular matrix. We use a couple of synonyms here. Sometimes you say underlying ECM, sometimes we say the underlying substratum. That's just another word that you might see for ECM. Sometimes we'll say the underlying basement membrane. Sometimes we'll say the underlying basal lamina. Again, you're building your dictionary. And here we have a different type of adhesion molecule. So we were talking about cadherins, desmosomal cadherins. Here, the protein mediating that adhesion between the cell and the underlying basement membrane, it's called integrins.
So for these anchors, the hemidesmosomes, is there some type of filtration process between the cell and the extracellular matrix? Because within cell-to-cell, it kind of makes sense where they want to be good, so they always keep themselves clean. But from cell to extracellular matrix, it's a lot different. So is there any type of
When you say filtration, what do you mean? it's not a hole.
Oh, okay.
Yeah, so he's asking if, in our cell-to-cell interactions, those two cadherins, are they kind of forming two holes? No, they're actually not like holes. It's more like this.
Okay.
Yeah, this was actually quite deliberate. So here's the cell, and these proteins are sticking out, through the membrane, transmembrane proteins. These are sticking out on the outside, similar over here, and they make contact with each other. So no, they're not holes. Yeah. They're what?
Anchor points.
Anchor points, yeah. Other questions? Okay. So we have integrins, and I'm going to tie this in with what Dr. Thung has gone over with you, by the way. All of this binding to cells, binding to ECM, critical in several ways. So I mentioned hemidesmosomes. Integrins are the adhesive protein. What are they going to bind to? There's no cell next to them with integrins also sticking out. These are binding the cell to the extracellular matrix, so they need to hold on to something in the extracellular matrix. And a lot of the times, that something is collagen. Sometimes that something is laminin. Not to be confused with lamins, which we find where? In the nucleus. Very different geographical locations. And students mix them up all the time. So laminin is very different from lamin. Okay? Had a hand? You good? Okay. Question? Okay. So integrins on the cell surface, laminin and collagen in the basal lamina. And there are other things in the basal lamina, in the ECM, and I'll tell you about some of those that hemidesmosomes can make contact with, whether directly or indirectly. So in the cytoplasm or in the cytosol, they're going to attach to intermediate filaments. Okay? Yes, question.
So the cytoskeletal is the intermediate filament, and the skeletal protein is the integrin. But then there are collagen and laminin.
Okay, so just to put them into context, she's asking about where everything fits. So the cytoskeletal component is one of our intermediate filaments, and that is connected to the plasma membrane protein, which is the integrins. And the integrins have to bind to something in the extracellular matrix, and that something most often is collagen or laminin. Yeah? Question?
Going all the way, like mitosis happening and the cell adhesion is going on. When will it stop? Like, when exactly?
When it looks like you. When will it stop, you're asking? When will all of the dividing and so on stop? When the baby is formed.
Yeah, but if,
And it continues as we grow.
okay. Like if you do exercise, like let's say, so the structure will expand, like, how would that happen? Like the cell division still, how they form?
Okay, so he's asking about, like when does this process of growth stop? I said two cell stage, we have adhesion, keeping those two cells together, and then 4-cell stage, etc., and you grow bigger and bigger. And then you asked about exercise in particular. Exercise is a little bit different. A lot of the times we're not necessarily making more cells, we're making our cells bigger. It's called hyperplasia. So that's a little bit different. But basically, as we grow, mitosis continues to happen for some cell types. And I think Dr. Thung will have told you about controls on mitosis, right? But let's say, okay, so a pregnant woman, that's easy. Her belly's getting bigger and bigger. And initially, it's just stretching, but actually, after a while, you actually have more epithelia being put down. The cells in the epithelial layer, they actually divide so that, because they can only stretch so much, right? So in that case, anytime we have mitosis happening to add to a tissue, you have the adhesion being required there. So it depends on where we are in the body when it stops, to answer your initial question. Yeah, I don't know if that helps. Okay. The story gets added to as we go along. Yes? Oh, okay.
Just so I can understand, this is the intestine, these are the cells,
Mm-hmm.
right?
Mm-hmm.
This up here is the
Microvilli?
apical surface,
Mm-hmm.
right?
Mm-hmm.
Okay, this is the blood vessel here.
Yep.
Bloodstream, I'm sorry.
Mhm.
Bloodstream. Is this like this now? Because in this case, this, you said the,
Oh, no. Okay, so he's asking about
see what I'm saying?
a schematic.
Or is it just the basal lamina here
No.
and then the bloodstream?
So let me just share with everyone. Okay. No, no, no. So in my schematic, well, if I can use this cell right here. So we're seeing this full cell and then a piece of these two, right? So his question was, I mentioned that somewhere down here we have the blood vessels. So clearly this is the apical surface. This is the basal surface. And so he was asking, is there another set of cells down here that ends up being the apical surface for these? Okay? And it's not like that. So this is one epithelial layer here, but the blood vessels have their own epithelial layer. So whereas this is the apical surface for these, go away, okay. When we go to our blood vessels, it has its own set of cells, and this is where they are.
Right, but what's in between? There's nothing in between?
Oh, there's a ton of stuff in between. I'll show you pictures later. All between here, components of the extracellular matrix. It's like one big mishmash of proteins and so on.
I thought glucose went from lumen to cell, and you can actually even have sodium then come out of the cell into the bloodstream.
Yeah, and I drew it as a space, but I kind of lied to you. I was keeping it simple. So earlier on when I told you about SGLT1 and GLUT2, I just drew it like this, and I said GLUT2 is down here, lets glucose out, and it goes into the bloodstream. But in real life, it's not quite that simple. It filters through there's this mishmash, I'm adding to the story now. It has to get through all of that and get in here, across that. Okay? All right. Good question. I'm liking that you're trying to put everything together in your head. Let me see. Let me finish up. Can I finish up hemidesmosomes and then we take a break? Yeah, let me finish up hemidesmosomes really quick and then we take a break. When you think about epidermis, when you think about skin, you cut yourself and you see blood, right? Even a paper cut. What you're doing is you're cutting through all of this. See all those layers of cells? You're actually cutting through that many layers of cells. And where are the blood vessels? Yeah, here are the blood vessels all the way down there. So this is just another, all of the stuff you saw on the previous slide, those are the cell layers there. And then what's below them? We have nerves, we have a whole bunch of blood vessels, we have a fat layer. So when you cut yourself, you're actually slicing through quite a few of them. So I just want you to get a sense. When we talk about cell to cell and cell to ECM, can you see the potential for cell to cell interactions here? So all of these cells have to bind to the cells next to them and below them. These last pink cells here, they have to adhere to these cells. And those have to adhere to these, and so on. These have to adhere. So there's a lot of cell-to-cell interactions going on. At the bottom, where we have and all of this is the epidermis. Down here, and it's kind of easier to show in this other picture, is the dermis. So epidermis, dermis. So when we talk about extracellular matrix, we're into the dermis. And I make that point. Anatomy is going to slow down and tell you all the gory details about these cell layers. But I bring it up now because I want you to make the distinction. If I ever, or if you ever, in a question, let's say, see something referring to the epidermis being detached from the dermis. Are we talking about cell-to-cell or cell-to-ECM interactions there? Cell-to-ECM, because the dermis, we are talking about the extracellular matrix layers. So the basement membrane, the connective tissue below that, all of that is ECM. So in this condition, again, it's an FYI, I'm gonna come. This is another skin blistering disease. And in this skin blistering disease, and again, an autoimmune one, we have antibodies being directed against some of the hemidesmosomal components. So if the cell now does not have a stable interaction with its ECM, we can have these blisters happening. Just by looking, you cannot tell the difference between EB and bullous pemphigoid. It's at the molecular level where the difference comes up. So in a question, when you see skin blistering disease, you cannot automatically think, oh, it must be something to do with the desmosomes. It could be hemidesmosomes, so you always have to pay attention. Are they talking about some cell-to-cell derangement or a cell-to-ECM problem? Okay, and I see your hand. Let me just, again, I'm pulling out a slide from the previous lecture that's relevant here, highlighting the important bits. Okay, question? Okay, all right. Any questions at this point? With so many proteins coming at you, you need a break. Let's do 10 minutes. Okay. Just a second. Okay, so I just made a call to see if I can figure out the color story, the color issue that I'm having. Let's see if, let's see what happens. So we finished off here. So let's move on to focal contacts. This is the last of our anchoring junctions. And the focal contacts, again, they're like the hemidesmosomes in that they mediate cell to ECM interactions, okay? They have different, oh, where am I going with this? I'm losing my train of thought. Oh, okay, I remember what I was going to say. So in terms of their major adhesion molecules, again, similar to hemidesmosomes, also integrins. But how do we distinguish them? The cytoskeletal attachment. So remember, for hemidesmosomes, it was intermediate filaments, but for focal contacts, the integrins in the plasma membrane are going to be attaching to actin in the cytosol. So let's talk a little bit about cell migration. So this slide, again, should be familiar. I think it's from that same cytoskeleton video. And it came up in the cytoskeleton video when we were talking about movement of a cell from one position to the other. So there's an infection somewhere in the tissue. The white blood cells in the bloodstream get a message saying, hey, we need you to fight. And then, so they go, they're moving along, you know, minding their business. But when they get that message, they slow down. They sit on the wall of the blood vessel, sit on those endothelial cells, and then they're able to work their way through that wall into the extracellular matrix. And once they're into the extracellular matrix, they can pull out their knives and guns and start killing off whatever bugs are causing the infection. Okay, so how do they do that? So again, starting from the top of the story, they're moving through the bloodstream, bulk flow. But they're going to stop at some point on the wall of the blood vessel, and they're going to start, instead of swimming along, being carried along, they're now going to need to start walking. And they're going to walk through the wall and get into the ECM. We talked about this because to walk, you need to rearrange what aspect of the cytoskeleton? Actin. Remember, we were talking about actin branching. That came up earlier. In this picture, so if you look at the very top one, it's like, so this is the leading edge, my front foot. I put it down. Notice what happens to my back foot. That raises up. Put it down. And every time, every stride, which is way too big for cells, but every stride is actin polymerization happening. When I put my foot down, though, what keeps it down? Integrins, focal contacts. It doesn't work if I have this foot suspended in the air. I'm going to topple over. I don't know how that translates to a cell, but it just doesn't work. So actin polymerization, so this part, this membrane pushes outward, put my foot down, integrins, focal contacts are going to keep that down. There are focal contacts back here too. But the thing about adhesion, most of the time, I think I have it on some slide, it has to be dynamic, meaning it's not like these two cells are sticking together and that's it for life. No, it doesn't work like that. They're dynamic. So at some point, those focal contacts are going to get a message saying, your job for now is done, let go, and that's how I'm able to raise up this foot. Actin polymerization, focal contacts. Focal contacts, let go, stop binding to the collagen, whatever. Polymerization, focal contacts. Are you getting?
Are you in the camera right now? See it later?
Yeah, I think so.
I don't know if it's
Oh, okay. I don't know, actually. I think so. I think so.
so the integrins help keep it active into the matrix.
Yes. She's asking if the integrins within the focal contacts are what will anchor my foot to the stage. My foot being the cell, the stage being the ECM. Yes. So leading edge temporally attaches to the ECM, blah, blah, blah. And So in this, the point above, this is not something to memorize, but again, tying things together. Necessary during embryogenesis, which again, you'll do a little bit with Dr. Thung. Movement of axons, nerve cells, in response to growth factors. So when easiest to explain that example with babies, you know, everything is formed from fresh, new, nerves are growing, figuring out what they want to do when they grow up. Do I want to go here? Do I want to go there? Oh, that one is sending me a message saying, come. So I'm going to actually grow towards whatever that trophic factor is. So it's saying that this is important in the movement of those towards growth factors. I mentioned the example of the white blood cells, and we talked about phagocytosis earlier. Any questions about this? Okay. So what two main properties do they have in common? Two main properties. Okay, so they have a similar cell adhesion molecule. Yep, integrins. Anything else? Connecting cells to extracellular, connecting the cell to the underlying extracellular matrix. Is that what I have? Yeah, okay, that's what I have. And again, you have these already, right?
Do the focal contacts with the integrins also do the laminin and collagen as well?
Do the focal contacts with the integrins also bind to laminin in the ECM? Yes, yes. I don't think I wrote that down, so thanks for asking. And collagen also, yeah. So someone asked me a question earlier. I told you, be careful about mixing up laminin and lamin. And she reminded me of a third one, the basal lamina. So laminin is a component of the basal lamina. Lamins are in the nucleus. Okay? And hemidesmosomes are stabilized by IF, intermediate filaments, excellent. And focal contacts stabilized by actin, good, or microfilaments, awesome. Let's do some more review. This stuff, you have to do it over and over again for it to stick, trust me. I had to learn it one day from scratch. Which one's a cell to cell and which one's a cell to ECM? Adherence junctions are? Mediate cell. So draw along with me on your slide, okay? All right. Cell to cell. What about our desmosomes? Cell to cell also. Focal contacts? Cell to ECM. So I press there for blue. Hemidesmosomes? Cell to ECM. I tried changing it here with the icon, which I can't see right now. I can change the color there, but this doesn't allow me to switch from one to the other. So as he helps me, let's move on to talking about the occluding junctions. So these were the anchoring junctions that did this, the occluding junctions. Now, when I do this, can you see me behind my hand? It's a silly question, kind of. But the point I'm trying to make is the function of the ones that we just talked about, they're for adhesion. And, you know, holding on to the cytoskeletal bit, strong adhesion. But if you poured some water through here, I'd get wet on this side. Ions can pass through here. This is the difference between the anchoring junctions and the occluding junctions. Now can you see me? Say no. You shouldn't be able to see me. The point I'm making is the occluding junctions are more like this. You poured water on this side and it doesn't get through. You try to get ions through, no. So occluding junctions, their main function is to form a seal. Occlude, to hide, that's what it is. So what are they sealing? Again, think of our two cells. I won't draw it right now because he's working there, but think of our two cells, two columnar cells, the rectangular shaped ones. Apical surface up here, basal surface down here, and this surface, the long side, what do we call it? The lateral surface. And we have our anchoring junctions, right? So there's actually a bit of a space, even though a lot of the times we draw them like sitting right on each other, there's space. And that space is referred to as the paracellular space. Para meaning, yeah, close, about, basically a space in between two adjacent cells. So our tight junctions, they're always going to be found closest to the apical surface of these two adjacent cells, and they're going to seal the paracellular space. Okay, what's so great about that? We're in the intestines. Be with me, in the intestines. And we've eaten some food. There was this little spot on that part that you didn't see that had some bugs in it, and that went down also. Ideally, that would be killed along the way, but your bad luck, it wasn't. So now those bugs are in your intestines. But we have our tight junctions. And we have our plasma membrane that's semi-permeable, not letting in things that shouldn't be in. And so in a perfect world, in a perfect bugs world, it would be able to simply get through that paracellular space. Why bother to go through the cell and deal with having to dodge lysosomes when you can just get in through the paracellular space? Huh. That's why we have our tight junctions. That's one of the reasons we have our tight junctions. Okay? And, you know, other simple things. We might want to regulate how much water is getting to and from all that basal surface. Is actually, so I'm filling in the blanks, and I told you, okay, so there's actually some interstitial fluid there, and then we have blood below and all of that. And some of that stuff also could potentially float up into the lumen. We don't want that to happen. Tight junctions. Sometimes, depending on what's going on, we may want to let in some water and ions that way. We won't talk about those scenarios, but you know, lots of things go on in the body. And so that seal can be regulated. But under normal circumstances, we don't need to use that. It's there, gate closed. So occluding junctions is supremely important for that sealing function. So, yeah, so let's just forget it. We're going to be with red today. It's not going to work for us. That's all right. So these are tight junctions. As always, there are proteins actually doing the hard work. Names of the proteins: occludins, claudin. Yeah, more names to remember. So the main TM transmembrane proteins that are forming that seal, occludin and claudin. And then just inside the cell, so these again, transmembrane proteins in the circles here, those are attached to these ZO proteins in the cytosol. ZO probably stands for zonia occludens. And then the ZO proteins in turn are attached to actin, as you see in the red box. Okay, so there's still a cytoskeletal component that they bind to. Zonular occludens. But that we tend to abbreviate. We call them ZO proteins. ZO1, ZO2, you're not responsible for the numbers. Just know where the ZO proteins are and know that they're an important component of that whole junction.
So the tight junctions is the paracellular space in the cytosol?
The tight junctions seal the space between cells, which is called the paracellular space. And they are plasma membrane proteins, they're membrane proteins. And sorry, what did you ask me about, claudins? Or yes, ZO is a cytosolic protein. I just want you to contrast it with the claudins and occludin, which are plasma membrane proteins, so that if I ask you, are they made along the secretory or non-secretory pathway? That's why it's important for you to know. So are tight junctions in the cytosol? No. They're points of contact between adjacent cells, just like our adherins and our desmosomes and so on. It's just that their function is different. Adherence junctions and desmosomes, their function was to form a strong attachment between adjacent cells. The tight junctions, they're not strong. They're weak as heck. But their function is to seal the space. So if we are trying to pour something down the space between the cells, the tight junctions are going to prevent that from going down. If we want to keep the two cells together and all we have is tight junctions, we're out of luck. I was going to put an adjective. We're out of luck because they're not strong. Tight junctions and zonular occludens, they are the same thing, but they're not a cytosolic protein. They're a junction. They're a I keep using the term, but because I can't find a better term, a point of contact between adjacent cells. So the main proteins that form a tight junction, they are plasma membrane proteins. They're part of the cell, but they're located in the plasma membrane. But those plasma membrane proteins, so if this is my lipid bilayer. Okay, and the rest I'll just draw as a straight line. My tight junction proteins, and I'm sorry I can't use a different color. My tight junction proteins are transmembrane proteins. They're embedded in that lipid bilayer. This part is facing outside, and hence that part is facing the outside, and okay. And so here would be the apical surface of the cells. Okay. And this part is facing, this part of the protein, this is the protein here. This might be occludin. This is extending into the cytosol. And this part that is outside, this part of the protein that is outside, that is making contact with another protein, the part that's sticking outside, on an adjacent cell that has run off our screen. And so I've drawn them badly. It's more like this. The protein on the adjacent cell is going to be more like that. So it's the parts that are sticking outside the cell that are making contact with each other to form the seal. Go ahead, formulate another question, because I see you're not quite satisfied. How does
the exercitis can happen? Exercitosis.
Exocytosis?
Can happen if they are like tightly packed, like the plasma membrane is tightly packed.
Okay.
Then where is the
So he's asking, with all of these proteins tightly packed like this, how do we get exocytosis happening? There are a lot of proteins on the surface of the cell, but then it's not completely packed with proteins. That's number one. And number two, if there's a region that happens to have a protein during exocytosis, well, it goes. The cell will make more. So think about it, that part, if a part happens to pinch off, maybe during endocytosis, let's say, you have that inward blebbing of the membrane, and there happens to be a tight junction protein there, the cell will make more of it.
So then all cells will not execute the exocytosis, like only some areas and some things happen. So
will all cells be able to execute exocytosis? Yeah, as long as they have, they'd have the capacity to do that. Might there always be a protein in the way? Not always, but if there is, no one's going to die. If one copy of the protein goes, the cell has means of being able to seal itself, seal back that part of the membrane really quickly. They've actually done studies to look at a nanosecond time scale to see, okay, if a part of this membrane gets pinched off for endocytosis or exocytosis, is there a big gaping hole? No, the cell is able to seal that really quickly. And again, if there was a protein there that got sacrificed in the vesicle formation, gene expression will make another one. Yeah. And it's not, don't think of it as, okay, so I'm a cell, there's a cell next to me, and there's this one claudin and one occludin. There are several of them. So we're okay. We're okay. Question?
So, between the two junctions we talked about, adjacent and oculating, which is structurally more stronger?
Ask that again, please.
So, from adjacent junctions and oculating junctions.
Adherence junctions, you mean?
Adherence junctions.
Yeah.
Which one is more stronger when it comes to like cell-to-cell interaction? Which one keeps the cells more together?
Okay, so he's asking me to compare occluding junctions and adherence junctions. Which one's the stronger? Not fair. Not fair. They perform two different functions.
Okay.
Occluding junctions, they're not strong at all. They don't have to be. That's not their function. Somebody else does that work. You don't ask your electrician to do your plumbing. Occluding junctions, they're there to seal. That's what they do. You might ask me, can we compare adherence junctions and desmosomes? Which one is stronger? That's a fair question. And I don't care, just as long as they both do their work. We need them to keep cells strongly adhered to each other.
Okay, sorry. So
Don't
you
be sorry.
mentioned that this is the tight junctions is a plasma membrane protein, but also a cytosolic protein. No.
So tight junctions also, both a membrane protein and a cytosolic protein? No. But the junction, it's a group of friends all working towards the same purpose. So amongst this group of friends are some plasma membrane proteins. There are also friends that are cytosolic proteins, but they're working together towards the same function. And they're together, collectively, they're referred to as a tight junction. No. Did I say there was cytosolic protein?
Yes.
I'm sorry. The claudin and occludin are transmembrane proteins. The ZO proteins are cytosolic proteins. The actin, cytosolic proteins. They are all connected to each other. The claudin and occludin are transmembrane proteins. They are connected to a protein in the cytosol, a family of proteins called ZO proteins. Those are connected to actin. Together, they're called a tight junction.
So we can automatically rule that this is a secretory pathway.
When you say this is a secretory pathway, no. No. Tight junctions are a family, are a group of friends. Some components are going to be made along the secretory pathway. Some are going to be made along the non-secretory pathway. Because two components are transmembrane proteins. The other components are cytosolic proteins. So the transmembrane proteins, those will be made on the secretory pathway. But the ZO proteins and the actin, non-secretory, because those are cytosolic proteins. Is that clearer? Anyone else? Are you good? Yeah, got it? Question? Wait. Go ahead, say again.
The difference between occludin and claudin where they are located?
So she's asking what's the difference between occludins and claudins, just so they're two different proteins. Not where they're located, they're both transmembrane proteins. They're both plasma membrane proteins. And they're both important. So if you say, oh, can we just take one out and still have that space sealed? No, you need them both. Yeah, but they're both plasma membrane proteins, which are transmembrane proteins. So if you look in the picture, the tight junctions are always closest to the apical surface. Beneath them are the adherence junctions. Beneath those are the desmosomes. This is not a random order. And no, you cannot interchange them. That is the order. That is why they say tight junctions help maintain polarity. Okay? So there's that word again, seal, regulates selective diffusion. So I tried to explain that with water and ions. It's a selective seal, so there are times when the cell might decide to open it up a bit, depending on the needs. We won't talk about those conditions right now. And as was just said, helps maintain polarity. just about every part of the body, excuse me, that we know of can be taken advantage of by bugs. So anybody ever heard of C. diff? You've heard of Clostridium? There's and a lot of them work similarly, but there's another one of that genus, and don't write this down. Clostridium perfringens causes diarrheal disease, just like the other Clostridia. And one of the ways in which that works, it produces a toxin that, in layman's terms, destroys one of these proteins, the claudins or the occludins. I can't remember which one. I think the claudins. So again, like I said, one component goes, and that abrogates that seal entirely. You need both the claudins and the occludins. And so in those patients, again, it's a diarrheal disease. So now that there's no working seal, water from the interstitial fluid on the basal surface of the cells, when I say water, I'm using the term loosely, but fluid, from the interstitial fluid, it can now exit through the paracellular space into the gut. And that's why the diarrhea is watery, because there's more fluid there than usual. And there are some other bugs that produce toxins that work in a similar way. Oh, for some reason that didn't come up. Okay, but we did talk about that. So this picture I showed earlier, and earlier we were talking about the adherence junctions, but we also have tight junctions, in our growing embryo, because we need to be able to control what's going through that paracellular space. It's always there. Okay, so gap junctions. You've heard about these before. Let's talk about them in a little bit more detail. So you know what they do, right? We talked about them in signaling, and I think in some earlier lecture also, cell-to-cell communication. What I may not have mentioned explicitly before is that they're a hydrophilic channel, so nothing lipophilic will get through gap junctions. Water, small ions. The moment we talk about a protein, not through gap junctions, too big. Size exclusion limit applies here, so about 1 kilodalton. So any type of protein, if that's, we're talking about movement of a protein and gap junctions are an option, just pass it right by, okay? Proteins are going to be too big to pass through here. So as the point says, direct exchange from one cell to the other. So in the picture, this is half a channel. This is one cell here, by the way. This is the membrane. Look at the lipid bilayer. And this is the adjacent cell. Now, in each of these cells, this structure right here, that is called a connexon. And it's actually, even though it's shown as kind of one thing, it's actually a multi-protein complex. And the proteins that make up the connexons are called connexins. So connexins make connexons, and connexons, two connexons together, constitutes our gap junction. If it's one half, it's not a gap junction. A gap junction provides communication between two adjacent cells. So again, cell number 1, adjacent cell number 2. I'm going to draw it a little bit farther apart just for effect. So this is a connexon in cell number 1, and this is the connexon in cell number 2. And so we can have things going through here. We cross the paracellular space because these two things kind of sit right on each other. So imagine a half a toilet paper roll in one cell and another half a toilet paper roll in the adjacent cell, and we line them up nicely. We get things through easily. That's our gap junction. And the most common example when gap junctions, you find them between all types of cells, epithelial cells, but a lot of the times we talk about it in terms of cardiac myocytes, where ions being able to pass from one cell to the other are what allows synchronous beating of the heart. So when you think about the heart, it's not big, roughly fist size, but think of the, thousands, millions of cells that make up the heart. And somehow they all work in concert to do the but-up that you hear when you put a stethoscope there. And it's on, picosecond type scale that those messages get passed from one cell to cell number 20 million in time for you to have this rhythmic beating. Gap junctions, they're cool, the coolest thing. One of the coolest things, I guess. All right, so electrical coupling, that's what I was trying to describe. And you find them between other cell types also. So there's always a condition, right? But again, remember, this is FYI. So this is showing you the importance. Actually, in one of Dr. Thung's lectures, I think it may have come up yesterday, he told you about syndactyly, right? Yes, he did? Yeah. So why do you have that happening? Because failure of apoptosis. And what tells a cell that it's time to die? Signals sent from one cell to the other in this region in particular. So in this particular condition, and again, you do not need to write this down, oculodentodigital dysplasia is the name of it. In this particular condition, there's a mutation in a particular gap junction gene. So the cells are not able to communicate because those proteins never got made properly. And so the ones that were supposed to die did not get the message. It's that simple. And so they didn't. And hence, you have syndactyly. It's less obvious here, but do you see it? So what should have happened, I guess, is that these cells here should have died so that it's on a similar level as that one. You always have to check, right? You know, like, I don't know if you ever did an undergrad psych class, and every time they describe a condition, you're like, oh, I do that. Oh, does that happen? Do I have that condition? So every time, I'm always like, so again, gap junctions. In this condition, there are lots of reasons why one half of your face doesn't look exactly like the other half, at least for a lot of people. In this condition, not saying that this is the case always, but in this condition, that's why you have the difference in the eyes, for example, okay? The ocular part of the disease. Sometimes there are missing teeth, the dento part of the disease, okay? So, yes. Uh-huh. Oh, no, it's missing teeth, missing. But in the boy above, yeah, that is a gap up there, but I don't know if maybe he's missing a tooth in general and it just shows up as a gap there. But in that condition, not a common condition, pretty textbook, you'll find those three types of symptomology. I'm not sure. I didn't read all the parts of the paper to say that this person in particular, whether he was missing a tooth and that's why you have the gap there, but I suspect that's what it is. Okay, so the CAMs in general, a lot of times you might hear CAM, cellular adhesion molecule. There are different ones that mediate cell-to-cell interactions, that mediate anchoring in general. And so you've heard about the cadherins already, but we're going to talk about the others. So you've noticed from the pictures, they all have a part that sticks out to the outside of the cell, the extracellular domain, and there's a part that extends into the cytosol. That's the cytoplasmic domain, because they have to bind to the cytoskeleton, right? Sometimes they bind to, some of them will bind to the same type of protein on the adjacent cell, so those are referred to as being homophilic. Sometimes they're going to bind to a different macromolecule on the adjacent cell, so those are heterophilic. So let's talk about all of those different components. We're going to talk about the cadherins in a little bit more detail from what we did before. So cadherins, when it says the primary cellular adhesion and cellular adhesion molecule in epithelial cells, it's just as it says. They're important for the adherens junctions. The desmosomal cadherins are important for the desmosomes also. So anytime you see cadherins, they're always mediating cell-to-cell adhesion. Now, remember that picture earlier on I showed you there's all this stuff in the cytosol that we won't worry about. Let's worry about it a little bit right now. So in the cytosol, what are the cadherins going to bind to before they actually bind to actin? A bunch of proteins in a family called catenin, C-A-T-E-N-I-N. Okay, so we're going to talk about one of those in particular, but just let's get the line up. First of all, we have the transmembrane protein, the cadherin. The cadherin has to bind to catenins, and then the catenins are going to bind to actin. They're all important. And by the way, the cadherins, you'll see somewhere mentioned that calcium is needed for its proper structure, so all of those things are important. If you take one component out, we're messing up that cell-to-cell interaction, that cell-to-cell adhesion. So let me tell you a cool story about beta-catenin in particular. And I'll go back to the embryogenesis story. So babies forming slowly, 2 cells, 4 cells, 8 cells, etc. You may have heard along the way, but if you haven't, don't worry about it. You've heard of cell differentiation for sure. So we start off with a ball of cells, but eventually we're going to end up with 3 layers of cells. I don't know, have you come across any of this anywhere in undergrad? When embryogenesis, 1 layer becomes 3 layers, and each of those 3 layers is going to become something different. Some will develop into the nervous system, some will develop into the viscera, etc. So let's say I'm in a ball of cells, and I'm a cell. This cell here is going to form another layer. So it's going to basically break away and migrate to a region just close by, and then it's going to start to divide. And it, at that point, is going to start to, quote unquote, look different from that initial ball of cells. So what has to happen? Remember I mentioned earlier, adhesion needs to be dynamic. It's not just like you're adhered here, this is where you're going to be for life. No. It has to say goodbye to its friends at that point, and it needs to migrate and then make new friends, and it needs to divide and all of that. So what's going to happen at a cellular level? There are cadherins keeping it bound to its current friends. One of the things that will make it leave that current friend, the beta-catenin, that kind of gets plucked out of there. I'll just use that term, plucked out, for now. So we're going to remove the beta-catenin component. It is going to relinquish its binding to the other components right there. And again, you take out one component, the whole deck of cards fall. So that takes care of its binding to its adjacent cell. But let's continue. What's the fate of that beta-catenin? So it can do a couple of things at that point. It's now a protein floating around in the cytosol with nothing to do. Yes. So it is going to be ubiquitinated by cytosolic enzymes, and then what's going to happen to it? Proteasomes. You make me proud. You're awesome. Proteasomes. It's going to be degraded because it's no longer performing the function it was meant to perform. But beta-catenins, they can do more than one thing. So let's say we're talking about the beta-catenin in that cell that is just detached. And that cell is going to grow up to become something. So that cell is going to need to divide to form that second layer. And so what happens there? The beta-catenin actually dodges around those polyubiquitination enzymes. The signal sent saying, not me, not today. And instead, they take a walk into the nucleus. And it turns out that beta-catenins are actually regulators of transcription. So they're going to go into the nucleus, and they're going to sit on some promoter region that is coding for genes that are involved in progression of the cell cycle. And so you have those genes being transcribed. What's going to happen to that cell? Mitosis. And that's how we're going to eventually form that second layer. So beta-catenins, the fates, there can be 2 fates depending on what's going on in the cell at that time. One, doesn't have any other function. Let's say it's a what's the term that Dr. Stung uses? A mature cell. Is that the word? Like a cell that's done dividing? A differentiated cell, that's the word I'm looking for. Let's say in whatever scenario, it's a fully differentiated cell. There's no more dividing that's going to happen there. It's an adult. So in that case, the beta-catenin, polyubiquitinated, chewed up. But if we're back in our embryo, and there is more differentiation to happen, cell layers to be formed, that cell needs to become 2 cells and become 4 and form us a nice layer. And so in that case, the beta-catenin goes into the nucleus, regulates gene transcription, promotes cell growth, cell division. Can you see how that can go wrong in cancer? And don't write this down, but in several types of breast and other cancers, they've identified that the problem, and again, there are different types of breast cancer, different etiologies, but in some types, let's say you have some epithelial cells and an epithelial cell detached from its matrix. Once it detaches, okay, that cell would normally die, but it doesn't die because its beta-catenin was able to bypass those ubiquitination enzymes, go into the nucleus, and here's a cell that was supposed to be dying, but now it's becoming 2 cells and 4, and that's the start of a tumor right there. Okay? But so anyway, that's an aside. The main point is for you to understand the importance of beta-catenins here. Okay? Any questions on that? Awfully quiet today. Okay. A lot coming at you. Oh, question, yes.
Let's say you have a calcium deficiency. What diseases can come from that, though, in this scenario?
If you have a calcium deficiency, what diseases can come from that? That's such a broad question. Everything from osteoporosis to... So in this scenario, if for some reason the calcium wasn't there, we would not have the cell-to-cell adhesion. It's that important. So all of these little things, cadherin, all of these catenins, including beta-catenin, actin, the calcium, you take out just one of them, and the whole deck of cards falls. Okay, so calcium dependent, that was mentioned down there. Immunoglobulin superfamily. So this is another type of cellular adhesion molecule. It didn't come up earlier in the junctions. Those junctions were like, you know, some of the major ones, but there are others. And so in some others, we have different types of proteins. Now, if you've done some immunology in a past life, you're familiar with the term immunoglobulin, different member of the immunoglobulin family. We're not talking about antibodies here. These immunoglobulins perform the function of cell adhesion. So what's cool about them? When I say they're homo or heterophilic, it means that the immunoglobulin superfamily member on one cell, again, transmembrane protein, can bind to an immunoglobulin superfamily member on the adjacent cell. Another option, immunoglobulin superfamily stuck in a membrane of one cell, can bind to integrins on the adjacent cell, and hence they would be referred to as being heterophilic. That being said, it's mainly cell-to-cell interactions that they're going to mediate. The adhesion molecules. So what are these? Immunoglobulin superfamily. It's a big family, lots of members. What are they called? And you're like, please don't give me another protein name, I'm sorry. Some of them, an example of an immunoglobulin superfamily member is ICAM. Another example is VCAM. There are others such as PCAM, and the names are looking like that. These are the two that you'll probably see very frequently. So these are examples of protein names within that family. Transmembrane proteins. In the cytoskeleton, they're going to bind to actin. So in the picture here, this is an example of a homophilic interaction where this immunoglobulin on this cell, in this case blood cells, endothelial cells, blood vessel wall cells I should say, are binding to an immunoglobulin on a platelet. Let's say we needed some blood clotting to occur. In this example here, we have the immunoglobulin on that cell, the platelets, binding to an integrin on the white blood cells. So you don't have to study this example, but it's just an illustration of the homo versus heterophilic binding.
The IPSF, would that be an example of antibody? The immunoglobulin superfamily, would it be an example of antibody?
No, immunoglobulin superfamily is in the same family as antibodies, but the functions are completely different. These folks here Their function is cell-to-cell adhesion. Antibodies fighting infections.
So these, the IGSF skeletal, cytoskeletal protein is integrin. Like the immunoglobulin is active.
No. So IGSF is immunoglobulin superfamily. That's what that means. And remember, any type of adhesion, cell to cell, cell to ECM, there has to be an adhesive protein. So the adhesive protein, the protein that is mediating the interaction, is either itself, immunoglobulin superfamily on one cell, binding to immunoglobulin superfamily in the other cell, or that immunoglobulin superfamily is going to bind to integrins. They're both transmembrane proteins. In the cytoskeleton, actin is what they're going to be holding on to. Okay. And the integrins in this case are what? The integrins are just integrins. The ICAM and the VCAM, those are immunoglobulin superfamily members. Yeah. So again, and I know this is a lot coming at you. There are these four major types. And so we're talking about number 2 now, and I'm telling you that sometimes number 2 binds to itself, just like cadherins are always going to bind to other cadherins, but sometimes number 2 binds to number 3. The integrins we've talked about, so in some cases integrins are mediating cell-to-cell adhesion, sometimes they're mediating cell-to-ECM adhesion. And so depending on the integrins, a detail that I don't necessarily want you to write down, but integrins, it's always 2 of them. So when you read, you see something about alpha, beta, alpha 4, beta 6, whatever. Depending on the alpha, beta configuration, those integrins might be mediating binding to an adjacent cell, or they might be mediating binding to the extracellular matrix, in which case they're going to bind to collagen and laminin. So that's why the cytoskeletal partner can be different. You're not going to be responsible for knowing, okay, is this one going to bind to actin or is it going to bind to the intermediate filaments? No, but I just need you to know that they can bind to either depending on where they are. Okay. And then the last of the 4 is selectins. So selectins are always going to mediate cell-to-cell interactions, but how they're different, one of the ways in which they're different from the others, they are mainly found in the bloodstream. So any question you see, and you see bloodstream, it's always going to be there somehow. Bloodstream, serum, plasma, something to do with the blood. The others are found there also, but the selectins are found only there. So if it's talking about adhesion between epithelial cells, it's not selectins. These folks are going to mediate adhesion in the bloodstream. Another cool difference about them compared to the others, so in all of the others, it's a protein on one cell binding to a protein on the adjacent cell for cell-to-cell interactions. Here, it's different. It's a protein, a transmembrane protein on one cell binding to a carbohydrate on the adjacent cell. That is different. That's a key difference where selectins are concerned. Protein binding to carbohydrate. And it could be binding to the carbohydrate part of a glycoprotein on the other cell or a glycolipid, whatever. It's the glyco that is going to be bound by the selectins. So we have these names LEP selectin because at first they were dealing with white blood cells, leukocytes, and they identified selectins. They're like, let's call them L-selectins. And then they found some on endothelial cells, the blood vessel walls, and they're like, these are the same but slightly different maybe. Let's call them E-selectins. And then they were looking at platelets, and they're like, selectins again, let's call them P-selectins. And then somewhere along the line, they're like, we can find P-selectins on endothelial cells also. And we're finding P-selectins on the leukocytes also, so they're all over the place. Don't worry about the L, E, and P. You don't need to distinguish them. Okay, so here again, restricted to the vascular system, we're talking about blood vessels. Cytoskeletal partner is actin. So you're going to make a table, okay? Make a table with all of this stuff that compares and contrasts these different cellular adhesion molecules, because you will be responsible for doing that on exams. Okay, so main function of tight junctions. What say you? Anybody? Huh? Seal. Okay, I heard the word seal. Good. Sealing the paracellular space in particular. What about gap junctions? What's their main function? Communication. Good. Something to say, communication. Remember, intercellular communication between cell number 1 and cell number 2. If I use the term intracellular, what does that mean? Inside the cell. And so talking about gap junctions there is irrelevant, right? Things within a cell talk to each other in a different way. Gap junctions, we're dealing with intercellular communication. And this one? The one we just did, okay, selectins. And let's take a break before our last half an hour, just a 5-minute break. Get through that. Okay, we're on the home stretch. Let's get back into it. So I want to start off this last part by telling you about the signaling tutorial that you have tomorrow. I put a document, I asked the course director to upload a document in tomorrow's folder. And in that, you'll see, it's a labeling activity. Do it beforehand if you come. It'll make the in-class session be a bit more, go a little bit more efficiently. If you don't get a chance to, still, they'll ask you to access it tomorrow. And sometimes students say, oh, labeling, that's for, primary school students. But it is just another mechanism for you to develop that muscle memory. You do need to know the pathways, you need to know the sequence of events, and that's why I put a simple, but if you go through it, and again, you have the document there, so you can fill it in and then you can erase it and do it again a few days from now to do that retrieval practice. It's important to know those pathway intermediates for each of the different pathways and not just know what they are, but know the sequence. Okay, so look for that in the folder for tomorrow. Okay, so in this last bit, we're going to do the last two objectives. For now, we're going to talk about MMPs, matrix metalloproteinases. So here's the lowdown. And forgive me if I go off slide a bit. So just talking about epithelial cells. Again, epithelial cells, they line a surface. The stomach, intestines, whatever. That surface needs to maintain its integrity. When I say maintain its integrity, it needs to be whole, the cells need to work, they need to form that surface. So even though we draw two straight lines a lot of the times, obviously you have the lipid bilayer, it's individual cells that those straight lines represent, just like those green cells up there. And a feature of those cells is that if they're going to be part of the epithelial surface, they have to stick to the cells next to them and they have to stick to the underlying basement membrane. Sounds simple, right? That's all we ask. Stick to the people next to you. Stay stuck. Stay stuck. If at some point they detach, are they helping us form our epithelial surface at that point? No. They're not fulfilling the requirement of staying stuck. And so, again, we were talking about cells earlier, but even in the body, we don't have places for hangers on. If they're not going to perform their function, what's going to happen to them? Okay? And that's normal. There's cell turnover. When I say cell turnover, I'm doing my job right now, I'm doing it quite well, but at some point I get tired, I die, somebody takes my place. Normal. So at some point, I'm tired, it's time for me to detach, and the normal course of events, apoptosis. And what happens to that space? Well, there are others ready to take your place, right? Always. Nobody's expendable, not even cells, in most organs. So mitosis happens, and those get replaced, normal turnover. So again, I want to emphasize, the moment I detach from that epithelial surface, stop sticking to the people next to me or to my basement membrane, apoptosis is what normally happens. Okay, so what do we have up here? So the fancy terms. Normal epithelial cells exhibit anchorage dependence. What does that mean? Just as I said, they have to stay anchored. And that is a requirement of epithelial cells. So that's what that first point is saying. And not only that, like I said, if one detaches, the others that are there, they divide, they make more. And so you're only going to be normal circumstances, okay? No disease state I'm talking about here. Cell proliferation is only going to happen if you are in place, if you're doing what you're supposed to do, sticking to the people next to you, basement membrane. Next point says what it says. You can read. Does that make sense to you? How is the presence of bound integrins inhibiting turning on of apoptosis? I just put this point in other words. Does that make sense to you? Given what I just described about staying stuck and all of that, does that make sense to you? Because you're anchored, the signals are sent, hey, saying, hey, that cell, it's doing what it's supposed to do. There's no reason to kill it. We're not murderers here, right? So, does that make sense? That second point now? Okay. If you detach, when you detach, point number 3, that's when those apoptosis genes get turned on. Make sense so far? Okay. So where's the whole MMP thing come into play? Matrix metalloproteinases. Just in an exam, like, what is that again? I learned so many protein names, I cannot remember. Where do those come in? Just look at the name. What does the name tell you? Matrix, what are we talking about? Extracellular matrix, that's what that's talking about. Metallo, there's some kind of a metal, don't worry about which one. Proteinase, destroys proteins, degrades proteins. So what do MMPs do? They are enzymes, protein A's, right? Anything with ASC at the end, that's an enzyme. They are enzymes. Do they digest fats? Carbohydrates? Proteins. They're enzymes that digest proteins in the extracellular matrix. The metal is there. It's usually zinc, but you're not responsible for that right now. Later on, maybe they'll ask you to remember whether it's zinc, magnesium, whatever, but there's a metal that's required, like a cofactor, but I'm not asking you to be responsible for that right now. But even with that, you can figure out what those do. So examples of MMPs, there are many, and sometimes you see MMP9 and MMP1, don't worry about the numbers. But examples of MMPs, if I tell you a collagenase, automatically, oh, that sounds like an MMP. Where do you find collagen? Outside the cell, as a part of your ECM. It's an MMP. We're going to talk about elastase. Elastin is a protein. That's coming up on a few slides. So if you have an elastase, it's an MMP that degrades that particular component of the ECM, elastin. Gelatinase, laminase, all sorts of ases. But they're usually going to have the name of that's similar to what they degrade. Like collagenase, they just call it collagens. Okay, so where do they come into this whole story? Regulate cell differentiation, proliferation, blah, blah, blah. Let's go back, where do we want to go? Let's go cancer. Because you did a little bit of that, you're going to do some more tomorrow. And don't write this example down, but I'll come back to that. So in some types of cancer spread from wherever they started. Let's say you started with a melanoma and then with a skin cancer and you end up with lung cancer. How the heck did that happen? Well, we started with a tumor. Maybe in some cases our beta-catenin, instead of being destroyed, it went into the nucleus and turned on the cell proliferation genes. So now we have a tumor here. That is the cause of our melanoma. And then at some point, those cells got some message or changed the message to be whatever they wanted to hear. They stopped adhering to the ones next to them and to the ones and to whatever is in the extracellular matrix, number one. Number two, apoptosis genes would normally kick in. They were able to dodge that. And number three, so now they're there, rogue, but they're trapped in this mesh work called the extracellular matrix. How am I going to get through that to get into the bloodstream and make my way to the lungs? They turn on those MMPs. Somehow those MMPs get turned on when they normally shouldn't. They start chewing up the extracellular matrix, and what does that do? Prevents a corridor for that rogue cell to get from here into those blood vessels eventually, and then be able to be taken to wherever in the body. That's metastasis in a basic form. So with that in mind, let's go back to this. So I didn't put the cancer example up here exactly, but I stuck a note there that is not on your slides. So I'm skipping the second point. What do you think the relationship is between PRB and MMPs. What is PRB? Yes, retinoblastoma protein. And what does it do in general? It's a tumor suppressor. Right, it's a tumor suppressor. So when PRB is working, do you think the MMPs are going to be working abnormally and chewing up ECM where they normally shouldn't? No. But when the PRB, which happens in some cancers, because Dr. Thang told you about that, right? When those, proteins stop working as they should, the PRBs, what do you think will happen with the MMPs? Exactly. So PRBs stop suppressing tumors, MMPs can get turned on and start chewing up. So then whatever tumor gets formed, partly because the PRB stopped working, now it has also helped pave its way to spread. And this is a true story. There's a lot of research that has been done on this. So that's why I wanted to make the link with what you learned with Dr. Thang. I didn't want you to stress about it too much. I didn't put it on your slides, but I'm just making the link for you there. Okay, so going back to the second point, so it can do all that stuff, cell differentiation, that it's important for all of those reasons, and we won't go into those mechanisms. You do that in pathology next semester. But the consequences for lack of regulation are what I just explained to you. Making sense? Okay? All right. Okay, so we move on.
When you, sorry, first question.
Uh-huh.
When you talk about the bone remodeling, are you talking about how maybe the osteoclast is eating up the bone and it's continuing, so that's how it's going to degrade?
Right. She's asking about bone remodeling. I put that there for context, but you're not responsible for that. You're not going to get into the mechanisms of bone remodeling and wound healing, I think, this semester at all. But don't throw your Med 1 notes away, right? They're important even up to Med 4. Okay, so summarize, we've talked about, I won't spend much time here, I'll skip through it fairly quickly actually, but we've talked about the epithelial cell barrier, cell polarity, apical basal surfaces, etc. I use a few examples with embryogenesis, so that's what it means by regulating cell proliferation in embryogenesis and also in other aspects of embryonic development. And again, you'll do a little bit more on that tomorrow with Dr. Thang. On this slide, I won't go through this slide with you, but I put this here for your study purposes in case it helps. There's a lot going on, so when you are studying, take a look up here, for example, at the apical surface. Read what it says and just make it make sense to you, okay? It all fits in with what we've discussed. So go through each of these, and I strongly suggest that you go through these, okay? Great examples here. And when I do the review with you on Friday, we'll do some questions that incorporate some of these principles. And we also talked about the 4 major cellular adhesion molecule gene families, right? Cadherins, immunoglobulin superfamily, selectins, and integrins. Let's talk about the extracellular matrix in our last few minutes. So I've been referring to it from practically the beginning, but here it is now. And in the picture on the right, that doesn't actually look busy enough to describe the ECM. It's this meshwork of lots of different types of proteins. So a lot more of these things going on here. Its functions are indicated there. When it says, substratum for adhesion, for example, collagen in the ECM is binding to our integrins, for example, on the membrane of a protein. Signal transduction, I'll touch on that maybe in the review, but it has several functions. The primary function is to help maintain the shape of a particular tissue. Amount and character depends on tissue type. You've done collagen with Dr. Barrick, right? Okay, so let me use that example here. The amount of collagen and the type of collagen helps determine the type of tissue. Example, collagen type 1. Good, excellent. When I say collagen type 1, you say bone, perfect. So the amount and character, the type of collagen helps determine whether we're talking about basement membrane of the kidneys or bone, for example. And there's a lot of collagen. So these are two more of those study type slides. Again, go through these on your own, see what's being said all down here in the figure legend, etc. So the people who have seen this before, don't say anything. I already see it's brimming. The others of you. What's the connection there? Anybody ever seen these movies with Sam Jackson and the one with Bruce Willis? Tell me really quick. Again, the people who have seen it before, I'm going to ask you, please, don't spoil my thunder. What happened in Unbreakable? Remember when, at the start of the movie, there's a baby born, and the baby was screaming its head off. And they couldn't figure out, I think it was a dark, stormy night or whatever. Have you seen the movie? Okay. And the doctor came, because the baby, it had been a few hours after the baby was born, and it was just screaming. It could not be quieted. And the doctor came, and what did the doctor say? Don't remember. It's okay. The doctor basically said, in dramatic fashion, every bone in the baby's body is broken. The little boy, yes, the little boy, where have you seen him?
On the commercial.
The commercial, the Shriners Hospital commercial. Osteogenesis imperfecta. That's what I'm going for here. That's what this, so in the movie, obviously not testable on exams, that's the condition that Samuel L. Jackson has. His bones are very fragile. I think he's basically in a wheelchair, and he'll say that, you know, he has had how many hundreds of fractures in his life, bone fragility. Anyway, I bring that up because that's a defect in collagen, a genetic mutation, as a matter of fact. So that goes to tell you about the function of collagen, providing tensile strength, meaning that when I do this, okay, making sure, my bones, I'm not screaming in pain. For him, for a real patient with the condition, no, you don't want to do that, right? Their bone will break that easily. So collagen helps provide tensile strength. Impart structure, yada, yada, yada. These are extreme cases, but they're showing you on the left having to put metal pins to try to straighten that leg. This is another, some other cases of collagen defects. And on the right, where the bone is really curved, not OI, but defects in collagen that will affect the structure and shape of the tissue, in this case, the bone. This is another condition called cutis hyperelastic. And again, true story, don't write this down. These are just anchor points for you, showing you the relevance of this. That man actually used to travel around in a circus because people thought it was funny that he could stretch his skin like that. His elastic fibers are fine, but the collagen, there's a defect. And the thing about collagen is, if you just have an elastic, you can stretch from here to thy kingdom come. If you have collagen, in layman's terms, that puts a limit on how much you can stretch. So his elastin fibers were fine, but his collagen was mutated, and therefore he was literally able to stretch his skin like that. And back in the day, they put people like that, unfortunately, in circuses. And so this is a different condition, Ehlers-Danlos syndrome. This will come up again in later semesters, and you'll learn more about the pathology. Okay, so moving on, we're going to do a fairly quick parade through a number of ECM proteins. The next one, well, this is a region of the ECM. I've mentioned it before, the basement membrane. It's described here in the first point. A lot of different proteins, two major ones being collagen and laminin, but they're highly cross-linked. By having a basement membrane around certain tissues, it compartmentalizes them. What does that mean? Here is the liver, and it is surrounded by basement membrane. Here are the kidneys, a separate organ, surrounded by basement membrane. So in that way, we have compartmentalization. Okay, we don't have mixing of kidneys and livers, for example. A physical barrier for the reasons that we discussed already. Shape, polarity, and Remember, we talked about collagen and laminin, two major components. They're going to bind to integrins in the cell of the overlying, cells. Laminin is the next one, so I just discussed, major component. Laminins will actually interact with the proteins around it, but remember that in our cell to ECM interactions, like hemidesmosomes, laminins are one of the major types of proteins that are bound by the integrins. And you can see this cross-shaped structure here, which you're not responsible for right now in terms of the structure, just its functions and its location. GAGs. So GAGs are cool. GAGs are what you find. GAGs and proteoglycans on the next slide. That's what you have at your elbow joints and your knee joints. What do you think of when you think of at your joints? What's that jelly-like substance? Cartilage. GAGs and proteoglycans are the major component of our cartilage. So what are they? They're polysaccharides. Everything else we've talked about so far have been proteins. These are carbohydrates. So polysaccharides, interesting about them, they have lots of charges, carboxyl, sulfate groups. These are big negative charges. And so all of our GAGs, those negative charges can attract positive charges, and an osmotic environment like that will attract water. Hence, the characteristic of our cartilage. So that when you do this, I'm not screaming in pain. I'm really banging myself up today. But anyway, you're not screaming in pain because the GAGs and proteoglycans within my cartilage have resisted compression. That's the way we tend to say it. They, like a shock absorber, that is what they end up doing. Know these names. So hyaluronic acid is an example of a GAG. It is an important one for you to remember. There are a bunch of others down here, and maybe I won't necessarily pummel you with those this semester, but these are also examples of GAGs. Remember hyaluronic acid at least. The proteoglycans are basically GAGs, but they have a protein core within them. So it's like the protein core, and these carbohydrates are attached to it. And that is basically the difference between them. This has a protein component, but the same things. Big negative charges attract water, help form that shock absorber function. So, and proteoglycans form what is called this ground substance, which is basically, so I told you about epidermis layer of cells, dermis. In the dermis, you have that thin basement membrane, and then you have the connective tissue, and somewhere in the midst there, you have this thing called ground substance, and the proteoglycans are a significant component. So you're seeing in the pictures some indication of that. Elastic fibers, so I mentioned those. Those are what allow the stretchability of the skin. You find them, so which tissue types? Think of blood vessels. Think of the blood vessels of the heart, for example. Every time blood gets forced out of the heart, and it gets forced out with quite a bit of force so it can get to all parts of the body. It's important that those vessels have elastic fibers so that they can stretch and then recoil when necessary. In terms of elastic fibers, these are the protein names that you need to remember: elastin and fibrillin. It was said that Abraham Lincoln had Marfan syndrome. I don't know if you've heard of Marfan. Again, I'm not going to ask you to diagnose based on signs and symptoms, but in that condition, persons tend to be very tall. They tend to have these, I don't know if you use the term double jointed, people who can do weird things with their joints, twist them in ways that the rest of us can't. So hyper-elastic joints, and they tend to have heart problems also. So you might see that in relation to Marfan, a mutation in the fibrillin gene in particular. Fibronectin. So these are, I've put glycoprotein here, but really they're all glycoproteins. Most proteins have a carbohydrate bit attached to them. So they're found in connective tissue. They can interact, so that you can find them in the basement membrane also, and they can interact with integrins on our cell surface. And they can bind to different components of the ECM. So some of the fibronectin will bind to collagen, some will bind to fibrin, some will bind to themselves. And then I think this is the last one, fibrinogen. So fibrinogen are notable for forming blood clots. And